biology and function of T cells, and cell-mediated immunity. Cell-mediated immunity is a function of T cells and a function of activation of T cells. Activation of T cells results in cell-mediated immunity. Okay, at first I want to talk about the TCRs. Do you know anything about the TCRs? T cells recognize the antigens in the cleft of MHC by TCRs. And this is the structure of TCRs that is similar to BCRs or antibodies on the surface of the B cells. And the TCRs composed of two chains, alpha chain and beta chain. And two domains, constant domain and the variable domain. Similar to antibodies. Antibodies contains constant domain and variable domain. Here in the TCR also there are constant domains and variable domains. And the TCRs recognize peptides in the cleft of the MHC by the variable domains in alpha chains and alpha chain and beta chain. And also there is rearrangement in the variable gene in the TCRs similar to BCRs. Can you remember the rearrangement of the variable region of the heavy chain and light chain? And segments of the genes that rearrange in the variable region? In the heavy chain, how many segments there are in the heavy chain gene? Segment D, J, J and V. Similar in beta chain is similar to heavy chain of antibody. And three different segments encode the variable region of the beta chain in TCR. Okay, with J. J segments, D segments, and V segments. And one J joined to the D, and after them one V joined to the DJ. And after them, the rearrange with BJ binds to the one segment that encode the constant region of the TCR, and after the mRNA transcript from CBET beta and the rearrange BDJ, and after the beta chain produced. Similar in the alpha chain. Alpha chain is similar to light chain of the antibodies. And there are two segments for encoding of the variable region of the alpha chain, V and J, and one segment for the one segment of gene for the encoding of constant region of the beta chain. And in this way, alpha chain produce and alpha chain join the beta chain, and this is TCR. Can you remember the different steps of the development of B cells? Stem cells develop to progenitor,
pre-B cells, then immature.
first pro-B cells. Pro-B cells and then pre-B cell,
then immature B cell, then
immature. Yes. Here the same, the development of the B cells, T cell, stem cell develop to progenitor T cell, and progenitor T cell develop to peripheral cell T cell, and after the T cell develop to double positive T cell. Do you know what is the double positive T cell? Double positive T cells express CD8 and both CD8 and CD4 on their surfaces. This is double positive that encode or express CD4 and CD8. And single positive or immature T cell. What is the meaning of single positive?
He'll have CD4 or
CD, yes. Express CD4 or CD8 in this stage. And after them, immature T cells develop to mature T cells. The maturation of the T cells happen in the thymus. These are thymocytes, and these are epithelial cells, cortical epithelial cells, macrophages, dendritic cells, macrophages. These are macrophages. The thymus, that all of these cells are involved in the development of the T cells. You can see the development of the T cells in this slide. T cells originate from the stem cells in the bone marrow, as you see here. And stem cell must be developed into the T cells, come to the thymus, to the blood. They are called common lymphoid precursors, and they come to the thymus. Here you can see T cell precursor. And T cells, these T cells develop to pro T cell and pro T cell, and in these stages similar the B cell development. In pro T cell, this segment join to the J segment in the beta chain. And after them, in the pre-T cell, the development of BDJ completed. The rearrangement of the BDJ completed, and here the T cells are double negative. What is the meaning of double negative? Versus double positive. Don't express, no.
Don't have a CD4?
CD4, yes.
CD8.
No CD4, no CD8. And after them, this double negative T cells develop to double positive T cell here. And double positive develop to the double single positive. And mature T cell, and mature T cells leave the thymus and enter the circulation, and it is ready for the encountering the antigens, immature T cell. What is the surrogate light chain in the antibody development? Is it important for the development?
Yes, it carries the
binds to surrogate light chain in the B cell development, bind to, join to heavy chain.
Yes, exactly.
IgM, moon chain. Okay. and induce some signals inside the cell, the B cells, and the signals tell the cell that do allelic exclusion. Do you know what is the allelic exclusion? What is the allelic exclusion? There are two alleles for each gene.
Yes.
In the development of B cell and the T cells, just one allele transcript
Yes. The other one.
encode. Just one allele encode. Paternal or maternal allele. And rearrangement, when the rearrangement is functional on one allele, the rearrangement on the other allele don't do. Similar here in T cell, beta chain expressed on the surface of the T cell in associated of a T alpha. Similar to, this is a surrogate alpha chain. And induce signals inside the cell, the developing cell, and this cell stops additional TCR beta chain glucose rearrangements or arrangement, allelic exclusion, and stimulate proliferation, stimulate expression of CD4 and CD8. Then after T cells, we have the double positive T cells. And cell becomes permissive for the TCR alpha chain glucose arrangement. And here the signal starts the rearrangements of the variable genes on alpha chain. Then development of the T cells happens in the thymus. The first step of development is development of stem cells to grow up. You are not into the class? No? Cortisol and then cortisol and double
positive,
positive,
single positive,
positive,
and mature.
immature and mature.
Ma.
Okay, mature T cells in the circulation are called naive T cells before encountering antigen. And naive T cells can recognize antigens well, similar to B cells. I explained yesterday for you. Just naive T cells or naive B cells just can recognize antigens well. Secondary lymphoid organs. Just can T cells, just naive T cells enter the secondary lymphoid organs, such as what are the secondary lymphoid organs? Which organs are secondary lymphoid organs?
Lymph.
Lymph node and spleen. Primary lymphoid organs.
Bone marrow.
Bone marrow and thymus. What are the functions of the secondary lymphoid organs? Function of secondary lymphoid organs.
Make a development for
B BCL.
Which is T-cell
T-cell?
development.
Yes. Bone marrow development of
BCL and
B cells
T-cell.
and thymus development of T cells.
T-cell.
And what's the function of the secondary lymphoid organ?
Activation for innate T-cell.
Yes. Encountering naive T cells and B cells by two antigens. Antigens present to naive T cells and naive B cells in the secondary lymphoid organs. Okay. And here, as you see here, naive T cells recognize antigens and the surface of dendritic cells and B cells and activate, proliferate and activate and develop into effector cells. What are the effector cells? Effector T cells. What is the effector B cells?
B cells make antibody.
Oh, and effector B cells, what is the effector B cells? Which cells is the effector B cell? Can produce antibodies.
T helper cell. T helper.
No, B cells. Plasma cells.
Plasma.
Plasma cells can produce and secrete antibodies.
Antibodies.
Yes, and plasma cell is effector cell of B cell, and effector cells of T cells are different cells. We have two subsets of T cells. Yes, CD for positive T cell and CD8 positive T cell. And these T cells, CD4 positive T cells, have different subsets with different functions. And these cells are effector cells after of CD4 positive T cells and produced after recognition of CD4, naive CD4 positive T cells. And these are T helper 1 cell. And T helper
2.
2 and
T helper 3.
3. T helper 17.
7.
3 main subsets of T helper cells. And these are effector cells because these cells can produce some cytokines to activate some other cells in the immune system. I'll talk about them in the following slides. And here, naive T cells in the lymph node recognize antigens, and after them they develop or become the effector cells. And these effector cells leave the secondary lymphoid organs, and in the site of infection, these effector cells can recognize again antigens and function against the antigens. Here, for example, here macrophages engulf the antigen, and these antigens inside the macrophages cannot removed without the help of the T cells, these effector T cells. And these effector T cells produce some cytokines and activated macrophages, and these macrophages, after receiving the help of the T cell, can kill the microbe inside of the macrophages. And you can see the effector cell at the site of in the infection eradicate the microbe. And naive T cells cannot help the macrophages in the site of the infection. Just effector T cells can help the other T cells in the site of the other immune cells in the site of the infection. Okay, you can see here. And the effector cell of the CD8 positive T cell is CTL. Have you ever heard the CTL? What is the CTL? Cytotoxic T lymphocyte. Cytotoxic T lymphocyte. And CTL is effector cell of CD8 positive T cells. Okay, this is the phases of T cell responses. CD8 positive T cell and CD4 positive T cells recognize antigen and activate. After activated, the most important cytokine that produced by activated T cells is interleukin 2. Here, interleukin 2. This cytokine is an activator of the T cells and has autocrine effect and paracrine effect. Can affect on itself the T cells that produce interleukin 2 and other T cells. Okay, after activation, the cells proliferate. This is called colonal expansion. And after them, they differentiate into effector cells and memory cells. Effector CD4 positive T cells and effector CD8 positive T cells and memory cells. And these effector cells, after them, can CD4 positive T cells activate the macrophages, B cells to produce antibodies, help B cells to produce antibodies, and can increase the inflammation. And also CD8 positive T cells can, effector of the CD8 positive T cells, or CTLs, can killing of infected cells, infected target cells, and can activate macrophages. MHC peptide, binding of MHC peptide and TCR cannot activate TCR itself. Other co-stimulatory molecules need for activation, binding of other co-stimulatory molecules is need for the activation of the CD8 positive T cells and CD8 and CD4 positive T cells. You can see here. One of the most important co-stimulatory molecule is B7 on the surface of APCs, you can see here, and CD28 on the surface of TC. T cells for activation should be received 3 signals. One of them is because of binding of the TCR and MHC peptide. Another one is because of the binding of co-stimulatory receptor, B7 and CD28. And the third signal is induced by cytokines. Interleukin 12 is very important for activation of the T cell. That interleukin 12 produced by macrophages or APCs. And another cytokine is interleukin 2, produced by T cells and has an autocrine effect and paracrine effect. Okay. T cells after activation, T helper cells, CD4 positive T cells, after activation become the effector cells, T helper 1, T helper 2, and T helper 17. What are the differences between the subsets of the T helper cells?
Cytokinase.
Yes, cytokine release. They are different in the cytokine release or cytokine production. What is the cytokines of the T helper 1? All of these cells express CD4, but they are different in the cytokines, produce cytokines. T helper 1 produce
IFN.
IFN,
Gamma.
gamma, yes. And responds to intracellular microbes. And in its role in the diseases, autoimmune disease, tissue damage and inflammation, increased inflammation and in chronic infection associated with chronic infection. And T helper 2 produce 4, 5, 13, yes, and response against healthy parasites and allergens. And its role in the allergic diseases. And T helper 17 produce 7. These are two isotypes of interleukin 17 and 22. And response to
Bacteria and fungi.
yes, extracellular bacteria. And have a basic role in the autoimmune diseases and inflammatory diseases. An antibody against the monoclonal antibody against IL-17 now used for the treatment of the autoimmune diseases, some autoimmune diseases such as psoriasis. You can see here the functions of the T helper 1 cells. Antigens present to the naive T cells, as you see here, in the case of MHC class 2. So, present antigen to CD4 positive T cells. And CD8 positive T cells recognize antigen in the cleft of MHC class 1. This is very basic. Again, CD4 positive T cells recognize antigens in the cleft of MHC class 2 or 1. One. CD4 positive T cells recognize antigen in the cleft of MHC class 2. And CD8 positive T cells recognize antigens in the cleft of MHC class 1. You have a short memory.
Yes.
How you can take the exam?
Yeah, for exam different.
Get
For exam different.
good. CD4 positive T cells recognize the antigen in the collect of the MHC class. Class 1 or 2?
2.
2, okay. And proliferate and produce interferon gamma. And interferon gamma is the most important cytokine for activation of macrophages. And without the interferon gamma, macrophages cannot kill the microbe in itself. The interferon gamma activate macrophages, and these macrophages. Did you have the class of innate immunity? Yes.
No,
Innate immune response.
not yet.
Yes.
محاضرة
Do you have the program?
سيلباس
No.
سيلباس
The-
سيلباس for the features of
Do you know anything about the innate immune system?
No.
2, 3, 6.
جدول عندي بس القديم ما عندي جديد. ما عندي جديد.
They usually don't come to the class with 6 students, from 34 students.
Yeah, unfortunately.
You know that if you be absent more than four sessions, you can't have the final exam. Yes. Okay. Interferon gamma also can activate B cells, and B cells produce antibodies. Antibodies can activate complement system and opsonize the microbes or neutralize the viruses. These are functions of antibodies. Do you know the functions of antibodies? No?
Attached to the microbe and recognize the macrophage to catch it.
Opsonization.
Yes.
And neutralization. What is the neutralization of viruses?
Viruses consumed by macrophage and it is
No, viruses have their target cells and have receptors for binding the virus to the target cells. Okay? And antibodies neutralize the virus, coat the virus,
so
and don't allow the virus bind to the target
To the
cell.
target cell.
This is neutralization. And opsonization facilitates the phagocytosis. As you see here, there are Fc receptors on the surface of the phagocytes, and
Attached.
that facilitates in this way the phagocytosis. Then two most important functions of interferon gamma. One of them activates macrophages for killing of microbes.
Microbes.
Yes. And activates
B cell
B cell
to
for
produce
production
antibody.
antibodies. Yes. And functions of T helper 2 cells. T helper 2 cells also when CD4 naive CD4 positive T cells recognize antigens on the surface of APCs, develop or become T helper 2 cells. It depends on the antigens that recognized by this naive CD4 positive T cells. And antigens determine that naive T cells become the T helper 1 or the T helper 2 or T helper 7. And after these cells, T helper 2 cells, produce some cytokines,
Like
4, 13, and 5. And you can see here the functions of these 3 cytokines. Interleukin 4, 13 can activate macrophages to activation macrophages and enhance fibrosis. You know what is the fibrosis?
When the tissues is damaged and
Proliferation of fibrocytes
yes.
and proliferation of
fibers.
yes. For tissue repair, fibrosis is a mechanism for tissue repair. After tissue, we have fibrosis after tissue damage. And another function of the T helper 2 cells by producing IL-5 is the activation of eosinophils to kill the helminths. Eosinophil activation, they granulate, and in this way they can kill the helminths. And interleukin 4 and 13 act on the intestinal mucus secretion and peristalsis. Yes, increase the smoky movement of intestine, and in this way can remove the helminths from the intestine in this way. And interleukin 4 affect B cells to produce IgE and IgG4, and in the human IgG4. And this antibody production, IgG4, remove the again helminths or protein antigens. And IgE are receptor on the surface of mast cells, and binding of the IgE and antigen results in the granulation of the mast cell. Then the functions of the T helper 2 cells are mast cell the granulation and increase the inflammation in the hypersensitivity reactions. And another function, intestinal mucus secretion and peristalsis, and eosinophil activation and macrophages activation. Functions of T helper 17 cells. Again here, naive CD4 positive T cell recognize extracellular antigens, as you see here, such as bacteria or fungi, and proliferate, and after them produce cytokines, IL-17 and IL-22, and these cytokines affect different tissues in our body. For example, here skin to produce antimicrobial peptides and increase barrier functions. Do you know what is the antimicrobial peptides that produced by keratinocytes of the skin, cells of the skin. And this production of antibacterial peptides increase the barrier function because these peptides can kill the microbes in the skin directly that can destroy the membrane of the or cell wall of the bacteria in the entrance of the skin, in the skin. And do you know any antimicrobial peptides? No? So you didn't have the class of immunology. You can see the program. Do you have the program?
We have skin immunity later. We have skin immunity in 2 weeks.
No, before. Before this session. Study more, please. You are medical students, you know? Okay. Okay. Defensin is an antimicrobial peptide. Defensin. And it can destroy the microbes directly, destroy the cell wall of, for example, some microbes.
Antimicrobial.
Gram-negative bacteria, cell wall of gram-negative bacteria.
Yes.
They can destroy directly the cell wall of the gram-negative bacteria. I-17 affects the leukocytes and tissue cells and B cells and skin or other tissues. And these cells induce this cell to produce chemokines, TNF. This is inflammatory. I1 and CSF. CSF, colony stimulating factors. And
TNF.
yes, these cytokines are inflammatory or these cytokines are inflammatory.
Yeah, increase inflammation.
Cytokines, yes. And these cytokines affect granulocytes or neutrophils and to increase the inflammation, neutrophil response. Then T helper 17 can increase the inflammation. Okay, you can see here cell-mediated immunity against intracellular microbes by CD4 positive T cells and CD8 positive T cells. From CD4 positive T cells, T helper 1 cells and T helper 17 cells can induce the cell-mediated immunity. As you see here, T helper 1 cells and T helper 17 cells recognize antigens in the class of MHCs. MHC class 1, 4.
Classic 2.
Class 2. Class 2, 4. Are you sure? Class 2, 4, CD4 positive T cells.
No, class 1.
Class 1, 4, CD4 positive T cells.
Class 2.
No.
Class 2.
No.
Are you sure? Class 2. Class 2.
Class 2.
Class 2, you? And CD8 positive T cell? Class 1.
Class 1.
Class 1, okay. Recognize the antigens and produce some cytokines for macrophage activation and inducing inflammation and killing of microbes. Then this is the cell-mediated immunity, activation of T helper 1 and T helper 17 cells to produce cytokines. And the cytokine produced here, what is the cytokine produced by T helper 1 cell? What is the cytokine produced by T helper 1 cell?
IFN,
IFN gamma. Interferon gamma. And by T helper 17 cell?
IL-6,
17. T helper 17 produce I-17, interleukin 17. And these cytokines can activate granulocyte neutrophils, eosinophils, and also macrophages. And another cell-mediated immunity is because of the activation of CD8 positive T cells, or CTL. CTL are cytotoxic T lymphocytes. What is the effector cells of the CD8 positive T cells? Effector cell. I give this question in the final exam, in your exam. What is the effector cell of the CD8 positive T cell? CTL cell. CTL, yes, cytotoxic T lymphocytes are effector cells of the CD8 positive T cells. Naive CD8 positive T cells become, after recognition of antigen, become effector cells. What is the effector cell of the naive CD8 positive T cell?
activation BCL,
No, what is the effector cell? Just name. What is the effector cell called? Effector cell of the CD8 positive T cell is called cytotoxic T lymphocytes. Or CTLs are the effector cells of naive CD8 positive T cell. And the effector cell of CD4 positive T cell.
T helper 1,
T helper 1 and 3 effector cells. T helper 2.
T helper 17.
These are 3 effector cells of CD4 positive T cells. And CTLs recognize antigen on the surface of tumor cells and infected cells, microbe or virus infected cells, intracellular microbe or virus infected cells. And after them, by some mechanism, kill the infected cells or tumor cells. Okay, you can see the steps of the CTL mediated lysis of target cell. Here, CT8 positive T cells recognize antigen on the surface of target cell. What is the target cell of the CT8 positive T cells? Tumor cells? Infected cells, virus infected cells, and tumor cells are 2 target cells of the effect of the CD8 positive T cell. And as you see here, antigen recognition in the clef of the MHC class 1 or 2 on the surface of target cell for CD8 positive T cells.
One?
1, yes. And CTLs recognize antigen, and here you can see the immunologic synapse. Immunologic synapse means that the binding of different molecules on the surface of target cells and CD8 positive T cells, as you see here. Binding of the MHC peptide and TCR, and binding of the co-stimulator molecules or activation molecules on the cells. And results in this formation of the synapse and recognition of the antigen on the surface of the target cells results in the granule exocytosis, or the granulation of the CD8 positive T cells, or CTLs. And this degranulation results in the death of the target cells. Okay, this is a picture of electron microscope, and you can see the synapse binding of the different molecules on the surface of target cell and CTLs. This is CTL, 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 and this is target cell. And this is the synapse and binding of the different molecules of the CTL and TC for killing of the target cells by CTLs. You can see here two mechanisms for killing of the target cell by CD8 positive T cells, or CTLs. One of them is perforin granzyme mediated cell killing. There are some granules in the cytoplasm of the CD8 positive T cells, and these granules contains perforin and granzyme. And after recognition of target cells or infected cells or tumor cells, the CD8 positive T cells release the granules, the content of the granules on the surface of target cells. And this granzyme and perforin, as you see here, can induce the apoptosis in the target cells and activate caspases. You know the caspases? They are enzymes that do the segmentation and cut the DNA to the segments, and in this way can kill the target cells or induce the apoptosis in the target cells. Then, by activation of the caspases in the target cells, they can induce the apoptosis in the target cells. And another mechanism of the killing the target cell in the fast ligand binding. CD8 positive T cells, or CTLs, express fast ligand on their surfaces. And target cell, what is the target cells for CTLs? What are the target cells for CTLs? Tumor cells,
and the virus
infected cells. Yes. And these target cells can produce fast on their surfaces, and binding of fast ligand and fast induce the apoptosis again in the target cells and kill the target cell. Then there are two mechanisms for killing of the target cells by CTX. One of them is
Can you repeat the question?
perforin, yes.
Beautiful.
Granzyme mediated.
And another one,
Fast.
ligand interaction. Perforin acts similar to C9. Can you remember C9 in the complement system?
Yes.
Okay. And what is the function of C9 in the
Makes hole in the cell wall and allows water
Yes.
into the cell.
Yes, to lysis.
Lysis.
Yes, to lysis. And perforin here acts similar to C9, and polymerization of the perforin on the surface of the target cell form a hole or pore on the surface of target cell, and this hole causes the lysis of the target cell. CD8 positive T cells also need the help of T helper cells, CD4 positive T helper cells, to activate. As you see here, this is CD4 positive T cell, helper T cell, and this is CD8 positive T cells. And CD4 positive T cells recognize the antigens on the surface of APCs, and CD8 positive T cells also recognize antigen on the surface of the APCs, and CD4 positive T cells produce some cytokines such as interleukin 2, and these cytokines can activate CD8 positive T cells to proliferate and differentiate. Then CD8 positive T cells need the help of the CD4 positive T cells. Do you know the CTLA4? CTLA4? No. Both the CTLA4 and CD28 express on the surface of the T cells. But CTLA4 expressed on the activated T cells at the end of the activation, at the end of the response of the T cells to the antigen. And both of these two molecules binds to B7 on the APCs. But CD28 molecule has the activatory function, activates T cells by binding to B7. But CTLA4, when bind to the B7, can suppress the function of the T cells, can suppress the activation of the T cells. This is the difference between CTLA4 and CD28. As you see here, the CTLA4 is used for suppression of activated T cells in autoimmune diseases, in the treatment of the autoimmune diseases. As you see here, CTLA4 binds to the one portion of immunoglobulin. When this CTLA4 immunoglobulin give the patient, this drug can bind to the B7 on the surface of the APC and prevent the binding of the B7 to CD28. And in this way can prevent the activation of the T cells, and in this way can suppress the immune response of T cells to the autoantigens. And this is a drug for autoimmune diseases. Can you explain for me the function of the CTLA4? What is the CTLA4? What is the difference The CTLA4 molecule beats CD28.
You could repeat it again.
CTLA4 is inhibition of activation of T cell.
Yes, yes.
But
Suppress,
yeah,
yes.
but the CD28 activation the
Yes.
T cell and kill, and the CTL can use
As a drug.
to
Yes.
immune response and stop the immune response.
Yes. Thank you. Okay, and this is quiz.